Good morning, everyone. I think we are just at the clock hour 12 p.m. It will take a few minutes to get started, but just wanted to thank you for all coming in person here and virtually. As you all know from knowing about this event, that this is a memorial lecture for my father, Ashok Gupta. This is the fourth run. Our speaker for today is Dr. Vikram Khurana, whom we'll be talking about. But I just wanted to take a couple of minutes to briefly talk about my dad, like I always do during this time. And then my colleague and my boss, Dr. Boyd, will introduce Dr. Khurana. My dad was born in 1956. This is the Hindi letters for his name, and this says that's when he passed away. So it was April 2021. So this is why this memorial lecture is held in April. And the funny part is April is also Parkinson's Awareness Month. Nobody planned for it, but that's how it is. And then, he was born in a rural area, sort of north of New Delhi, like maybe 10 miles, which is where I was also born. And as you likely read through his brochure, he was one of the four siblings, and me and my two siblings were his children, and he raised a very extensive family during his lifetime. He got quite educated, coming from a rural background. He raised, he got multiple degrees. He served as a school teacher for 30 years. Came to Delhi when I was like in 5th grade, gave us all of the education we needed, as well as the, values. Really was a dedicated teacher. I still remember, retired in 2015 and lived with Parkinson's for 13 years. He was diagnosed when I was in final year of medical school, as you read in the brochure. I always feel that he was a life worth lived for over 4 years. And I always like to say one of the main reasons for me to have established this memorial lecture has been to feel that he's visiting me, because he never could visit me in his lifetime to the States. So that's one of the biggest factor and motivation for me. With that said, I will now pass the torch to Dr. Boyd, my colleague, to talk about briefly Dr. Khurana, our visiting speaker. Sorry.
Good morning, everyone. Thanks for coming. And thank you to Dr. Khurana for joining us. It's really an absolute pleasure to introduce him. Dr. Khurana is really a leader in the field of Parkinson's disease and neurodegenerative research. He has gone the path of coming to the United States from first doing his medical degree in Sydney, where he was born, and then came to the US to complete his research training. He did his MD in Sydney, came here to do his PhD at Harvard, stayed on to do residency at the Brigham, and has been on the faculty there since. His areas of research, he's really been a pioneer in our understanding of alpha-synuclein protein misfolding and some of the influences both of environmental as well as genetic factors and inflammatory factors. His approach, which is incredibly unique, and I'm hoping we're going to hear a lot about it today, of what he describes as precision medicine and working with pluripotent cells from skin fibroblasts to identify specific areas in which there may be opportunity for customized precision medicine for an individual based on what their genetic makeup and active factors in terms of their neurodegeneration are. So I won't take away too much time. As we had our conversations this morning, we kept asking questions, and he was wondering whether or not he should add more and more slides. So I'm hoping that I give him enough time to talk. So I won't take any more time. And thank you for joining us, and we really look forward to it.
Okay, thank you so much for that kind introduction. Yes, prepare yourself for a 4-hour lecture of all these slides that I added during breakfast. I'll try not to damage you too much. Thank you so much for the invitation, Deepak. Very, very a warm occasion, and I'm really honored to participate. Thank you, and thank you for the kind introduction. So I have disclosures that I hope will not affect the quality or integrity of my talk. I have started a couple of biotech companies, and I will be mentioning on occasion some of the work that we've done there. But all of that work is preclinical and not clinical, and I don't see any conflict there. But I will call that out when I talk about this. So I'm going to start with what I call the iconography of Parkinson's disease. You know, when we think about it, we think of, you know, patients like this woman is moving very slowly. She has executive dysfunction. She has exhibiting in this video the classic features: slowness, rigidity, tremor, instability. You've been to these lectures before. I don't know that you're all Parkinson's experts, but I expect you're all medical or in medical science. And so probably this is not new to you, seeing patients like this, and you're accustomed to these kinds of features of the Parkinsonian syndrome. And then, of course, when we think about a patient like this, we think of dopamine. I know you've had lectures in the past from experts who have told us that there's dopamine depletion in the brain, and of course there is. And it is because that dopamine itself, levodopa, is such a profoundly effective medicine that we have tied Parkinson's disease to this dopaminergic iconography and these movement disorders features that really tie to dopaminergic dysfunction. And then many of you will know, 100 years ago, a little bit more than that, one of Alzheimer's students, Friedrich Levy, described this very interesting lesion in the brain. You can see it here in a single dopamine neuron, this ringed structure here that is called a Lewy body. And we think of alpha-synuclein, this protein that clumps up in amyloid fibrils. And this, for me, is the iconography of Parkinson's disease: a motor syndrome related to dopamine neurons, and related to the misfolding and aggregation of the alpha-synuclein protein. A very linear story, and still the one that most people carry around in their head, including people who are making drugs for this disease. But for those of you who see patients with Parkinson's disease, you will know that this story is really incomplete, and we need to make it more complete in order for us to really effectively understand the disease and treat our patients. For example, this is not just a disease of dopamine neurons. This protein, alpha-synuclein, is found throughout the central and the peripheral nervous system. These days, we can capture synuclein from a deep nasal swab. We can capture synuclein from a skin biopsy, from the spinal fluid. Synuclein is in the nerves that innervate the heart, in the gut. It's affecting bladder function, bowel function, sexual function, blood pressure regulation. This is a diffusely present protein that I personally think is sitting at the interface of the environment and the patient, because it performs extremely important functions in stress responses. It's just doing its job, but beyond reproductive age at 40 or 50 years old, beyond that time, it can have maladaptive effects in the brain, and we can talk about some of those. So it's a diffuse disease, but it's also a really heterogeneous disease. When we see patients with this disease, our patients are clearly not created equal. This is real data from the Michael J. Fox Foundation Parkinson's Progressive Initiative, PPMI, and Markers Initiative. And this is real data. Many of our patients over 5 years, you know, a third of them hardly progress at all. And you see those really kind of sitting towards the bottom of this curve here, and others really dramatically progress over this period of time. What underlies this heterogeneity? We need to develop a way to systematize our approach to understanding, how did you get here as one of my patients, and where are you going to go? You know, we talked about this just an hour ago. What's your trajectory going to be? How am I going to predict that? And are you all molecularly the same, or do you have different underlying biological principles that are governing how you got here and how you're going to progress? Because an effective disease-modifying therapy is going to need to take that into account. And it's not just it's not just motor data. This is motor data here, but it's also cognitive data. This is cognitive data from our patients. Also, these ones happen to be in the PPMI cohort as well. We have a similar cohort that I discussed this morning, the Harvard biomarker study, where we also track patients and we see very similar heterogeneity. So what's the source of this heterogeneity? This is a complex question, but I'm going to break it down to the three things you see at the bottom left here. Complex genetic background, environmental exposures, and different conformations of this protein alpha-synuclein. So we tend to think of it as a static protein that forms these aggregates, but actually it turns out there's a whole menagerie of these aggregates. There are many different ways this protein can misfold, and we need to incorporate that biology also into the way we think about our patients. Now, I can't cover, we have a lot of projects in these different areas. I'm obviously going to just cover a subset. I think my slides are advancing by themselves, which is interesting. I might have to change the mode in a second. But I'm going to try and tell you a story about each of these three things, and we'll see how far we get. If I need to gloss over a few things, I will do that. But the first story I'm going to tell you is a story about genetics and our approach to trying to figure out if our patients belong to different molecular subtypes through their genetic background. So this is a snapshot of the history, actually a 5,000-year history of Parkinson's disease. I'm not going to go through it, but the disease has been described in various literatures and oral traditions for a long period of time. There was even a dopamine-containing herb that was prescribed for it around 600 BC that some patients of ours still take, Mucuna pruriens. But it was a seminal paper in 1817 by James Parkinson that really described in a spectacular way the precise features of the Parkinsonian syndrome. Now, if we flash forward to the 1980s, in the 1980s, people thought Parkinson's disease was purely an environmental disease. And that's because families with this disease were really not described. And there were some really striking examples. For example, people having contaminated intravenous drugs that would allow them to come in acutely with something that looked like Parkinson's disease. And of course, there was dramatic responses to dopamine. So people thought this was an environmental disease. Now, in the 1990s, one of my senior colleagues, Larry Golby, who was working in New Jersey, started to observe that, you know what? Parkinson's in some families seem to exist in every generation. And so through that painstaking observation, we started to find women like the one I showed you right at the beginning, who harbored different alpha-synuclein mutations tying the pathology, this alpha-synuclein pathology in the brain, once and for all to the genetics. So we started realizing across these diseases in the 90s and the early 2000s that you could just inherit extra copies of this gene and you would get this disease early, or you could have a point mutation in this gene encoding synuclein and you would get this disease early. And so these pedigrees started to be established, the synuclein gene was identified, and it was the identification of that gene that actually led to the biochemical identification of synuclein as the principal component of the Lewy body. So this was a really interesting time, and it was around this time that I joined Mel Feeney's lab as her first graduate student. She's a Harvard professor who showed that you could just put extra copies of synuclein in a fruit fly and it would stop climbing and it would develop Lewy body-like structures. And she, really paved the way for using model organism genetics to understand the biology of this disease. Fast forward just a couple of years, and my postdoctoral mentor, Susan Lenquist, said, this is an ancient problem, protein folding. I bet I can put the Alzheimer's and Parkinson's protein even in a simple yeast cell and learn a lot about the basic biology of what these proteins do. And so this was an exciting time where we knew that extra copies of the protein caused the disease in people, and extra copies were also causing toxicity and interesting phenotypes across model organisms. Now, if we fast forward to the present time, a number of genes have been described that have different levels of penetrance, different levels of severity, are alleles in the population that are expressed at different frequencies, but they only currently explain 10 to 15% of Parkinson's. And one of the reasons we don't have a better sense of I think the genetics of this disease, although these results have been obviously pivotal to our understanding, is that genes work together, proteins work together. So I'm going to tell you a story about how we think of networks of genes and proteins and not just individual players. And that gives us a lot of power to find genetic subclusters within the population that we think are going to be important for developing personalized medicines. And so where we start with this, of course, is let's start simple and see if we can design drugs for the simple diseases where we know the mutations. And we have been very focused on the alpha-synuclein gene itself, but other people have had focus on some of these different areas. Now, as you mentioned, as was mentioned in the introduction, there's a strong emphasis in my work in human stem cell models. When I was a junior resident at MassGeneral and the Brigham, Shinya Yamanaka, a Japanese researcher, published a stunning paper, some of you will be familiar with it, where he showed that we could take a skin cell from a mouse and then eventually showed it in human and just put a few transcription factors in it, turn it back in biological time, and it became something that looked like an embryonic stem cell, and we could then pattern it into different cell types. So this was remarkable for me because you see, at that time, we only had postmortem brain, but this was an avenue for us to be able to finally get a glimpse at how these diseases unfold at an individual patient level in the dish. But of course, this came with its challenges. So this is the basic idea. We make these, we take a skin biopsy, we turn it into these iPS cells, induced pluripotent stem cells, and we can make the very same dopamine neurons that we know are succumbing in this disease, or cortical neurons, autonomic neurons, you name it. Having said that, these are really immature cells. This is a disease of aging. So at the time, and this is really a principle that has governed my entire career to date, at the time, the big question was, forget it, don't make stem cells from patients like this with diseases of aging. You're not going to learn anything because you can only age them for a little while. They're immature cells. So, and it was a very real concern. So my entire career is really summarized in the aphorism of finding needles in haystacks. How can you take a patient's stem cells? I could make stem cells from all of you here. They would all have different properties. Some people may have some condition. How would I know that all of this random differences that I see in the dish are actually related to the disease? That was a real challenge at the time, and we spent 4 or 5 years addressing it in 2 very different ways. The first is we found some of those last surviving members of Larry Golby's cohort in New Jersey, and we made stem cells from them. We knew they had an alpha-synuclein mutation, and we genetically corrected it so we could compare apples with apples. We would have their stem cells and their stem cells with that single change converted back into the normal form so that we could now compare and see that difference is going to be related to the disease, right? Because it's a very specific change. Some of you have heard of genome editing like CRISPR, I think, but there was no CRISPR at that time. There wasn't even talons. There was zinc fingers. So it took us 2 years to make this genetic correction, something that we can do a month. Takes a month now, took 2 years. And then, even then, you have these cells, so they're really pretty and they're cool, and you get a nice paper out of them, but what do you look for? So Susan told me, you know what? Why don't you express, chop and change every gene in the genome of this simple cell yeast, put the same protein that's malfunctioning in these patients in this yeast cell, chop and change every gene in the genome, and start screening hundreds of thousands of molecules to figure out what makes these yeast cells better. And so with a large team of people, including Chi and Dan, who are pictured there, and they're my two mentors, Susan on the yeast side, Rudy Yanish at MIT on the stem cell side, we started to build this platform across a number of different diseases, including Parkinson's, and something really cool happened. We started finding yeast genes that got them better, and it turns out they were known human Parkinson's genes. So we did the same for Alzheimer's, and we found Alzheimer's genes. Aaron Gittler, one of my colleagues, discovered new ALS genes from a yeast screen. So it was a really cool time where we realized that some of this biology is conserved across billions of years of evolution, and we can use this biology to find a needle in a haystack in these very complex human neurons we were now growing from our patients. So at that time, I took a little sidestep and I started the company Humanity with Susan. I'll tell you a little bit of a story there. So as you increase synuclein levels in a yeast cell, they start to die. And they actually form inclusions that you can see in green here, these little bright dots, a little bit like what we see in humans, except they die before they get amyloids. They die when they start clustering vesicles. Synuclein binds synaptic vesicles, and even in yeast, it binds vesicles. And so they started to clump up and cause these yeast cells to die. Turns out, and I'm not going to talk about this work today, but we had a recent paper. I should say that, of course, as yeast cells have increased copies of synuclein, they die. And we know people succumb to Parkinson's disease and dementia, like the woman I showed you when she had extra copies of synuclein. And what we did then with CRISPR and zinc fingers is we made those genetic corrections. And over the years, remarkably, we have found that the same pathologies we find in the human neurons, we can find them even in a yeast cell. You've heard Parkinson's is probably related to mitochondrial dysfunction, happens in yeast, reactive oxygen species, reactive nitrogen species, disturbed protein trafficking. Susan was the first person to publish that vesicle trafficking was affected in Parkinson's, and now, of course, most Parkinson's genes fall in this pathway. So, and more recently, my lab has published papers on perturbed gene expression and mRNA metabolism. It's really very interesting that we could find quite a lot in such a simple cell, but the human neurons and glial cells that we were making were very validating. They told us that we were on the right track. Now, these types of inclusions, these lipid-rich ones and these fibril-rich ones, these are just not a yeast epiphenomenon. We have recently shown and built on a literature that's really 60 years old from Japan that these inclusions also form in the brain and that we can capture them in human stem cells. So this is a recent paper that we had. I'm not going to talk about it today. It's published, but we can basically capture within 2 weeks now a high complexity of alpha-synuclein inclusions in patient stem cells that look very much like the diverse inclusions we see in the brain. So we can have a much more nuanced understanding of what this pathology is. So we started this company and we could screen hundreds of thousands of molecules in this organism. This is an example of a molecule that can really protect these yeast cells. You see they start growing better with it, but it also kills them at really high doses. We use that property and the power of yeast genetics. So here you can see we take a toxic level of the molecule. We kill all these yeast cells, but you can see 4 yeast colonies are surviving. We sequenced their genomes and we found that they all had the mutation in the same gene. And that was now the drug target. So I'm cutting a long story short here, but we found the first lipid-related target for Parkinson's disease and developed it into a drug. And one of the things we did, what it does is it changes the fluidity of membranes and the relationship of alpha-synucleins binding to those membranes. But I'm not going to go into molecular details today. I'm going to just say that this molecule not only fixed a yeast cell, which would be rather boring, but we made these 3-dimensional organoids from these patients. These are now little mini brains, and you can see this abnormal form of synuclein is accumulating in the bottom one where it's her cells, which have 4 copies of synuclein, which is abnormal, and not accumulating in the top one where we've genetically corrected it to 2 copies. And the drug that we made can reverse this defect, and there are lots of changes in fatty acids and lipids that occur in these models, and they are also reversed by the drug. So the idea was that we took a simple patient with with a known mutation. We made her stem cells, and we created a platform that allowed us to rapidly find a drug. And we did it for relatively cheap. It normally costs billions of dollars to make a drug. We did this for around $100 million. And instead of taking 10 to 12 years, we took about 4 years. And so we got it into patients within around 4.5 years. And this drug is now being optimized by the company Janssen, which is the pharmaceutical wing of Johnson & Johnson. So that's an example. So we made a drug, but the question is, who do we give these drugs to? And that takes me back to the original question of the heterogeneity of these diseases. Parkinson's patients are not created equal, so do genetic subtypes exist? And so if I consider some subset of the Michael J. Fox Foundation cohort, I told you that some of these patients are progressing super fast, some of them are progressing not at all. Can we find the underlying genes related to this? And so these are really the questions, what are the factors that drive these outcomes? And is disease risk the same as progression? And can we ultimately find drugs that turn a fast progressor into a slow progressor? Now, one of the things that is invariant here is that whether it comes early, late, or somewhere in the middle, the protein alpha-synuclein is involved in this disease. Each of these patients at postmortem will have synuclein deposits in their brain. And so what we do, and I'm going to show data, but I want really just the concept to come across, what we do is we create a roadmap, let's say, of every genetic and physical interaction of this protein as it misfolds, and that is the search space in the genome where we look for clusters of genes that might associate with these different outcomes. And so that's a visualization of this diverse protein. And then we've got all of these diverse strains, these different shapes. But really, this protein aggregation property is happening in all of these patients. But it can't be all that is happening. And we started, so when I was a postdoc, we started making stem cells from single patients with mutations. But when I started my lab, I found an entire kindred in northern Spain that I now follow, which they have an alpha-synuclein mutation, and every generation, there's very variable outcomes for these patients. Some of them also progress very fast, and some of them really don't get the disease at all. So we, for the last 5 to 10 years, have been studying these patients, and they taught us one thing. They taught us that just having a mutation in this protein is not sufficient to get this disease. Other things have to happen. And so that's what if alpha-synuclein is represented as that green jigsaw puzzle piece, there's a whole lot of other stuff going on. And this is an example of 3 of the younger members of the family. And you can see, whoops, this keeps flipping. It's okay. The family here, members, this young person is completely unaffected. He's actually the oldest of the lot, but his cousin and his brother's younger, they all have the same mutation. They have Parkinsonism and dementia in their 30s, whereas he's completely asymptomatic. And this happens in every generation. We see these differences. So more than synuclein is involved here because they all have a synuclein mutation. So we're trying to hunt for different factors that might be related. And the idea here is that when a protein misfolds in a very tight space in a cell, it affects a lot of other proteins around it. So again, I'm not going to bore you with the details, but what my lab does is we take different models. They could be yeast cells, they could be brain organoids. We could do this in the human brain as well. We start to map out what does synuclein interact with, and how do those interactions get screwed up when it starts forming these aggregates of different description? And I'm just representing it here almost cartoon-like, that you might hear people say, is this a loss of function disease or is it a gain of toxic function? That doesn't make a whole lot of sense to me. It's both. There's toxic things that happen when these proteins misfold, but there's also places where these proteins should be, and so they are loss of function diseases as well. And we try and map those out, and that's what I'm really representing here. We map these things out with systematic genetics and systematic biochemical or other types of proximity labeling mechanisms. And so what you end up having here is a map of factors that are affected by the protein as it starts to misfold. And what I visualize is that on these maps, our different patients live in different corners of our maps. Does that make sense? That there are lots of connections to these proteins, and some patients come from one direction, other patients come from another direction. The treatment for those patients may be very, very different. And we do this, as I mentioned, in different cell types. And so the idea here is that people, whether they're in the family that I study in Spain on the left, or they're in the Michael J. Fox Foundation cohort, they live in different parts of these maps. So, and that's the hypothesis I write up here. And so the hypothesis here is that we should be able to find mutations or clusters of mutations in corners of our map that are kind of representative of subtype 1, subtype 2 of these diseases. So for years, we've been producing these maps and validating them in human stem cells and doing a bunch of stuff, and they're listed in this paper. But you know, this type of map is what I really describe here on the bottom right. You can see the synuclein protein here, SNCA, and it is linked to hundreds of other proteins in the human proteome. These are not in silico predictions. These are all from our painstaking experiments and then validated, where we look at every protein in the cell, and we basically chart out how these proteins interact with each other. And here are the membrane trafficking proteins. Here are the mRNA metabolism. And yellow is calcium, autophagy on the top right, oxidative metabolism on the top left, and so forth. So these are different pathways that people have tied to the disease, but this is how they all fit together, okay? And this is kind of really key. And this is Samaya and Xinyuan, who do a lot of work either in statistical genetics or using the stem cell models and manipulating all of the genes to create maps like this. So this is the concept, that our patients have 3 different phases: a risk phase, an onset phase, and a progression phase. When we see them in the clinic, they are progressing, and that biology could be completely different to the risk biology. So if you're designing a drug for someone who walks into your clinic, you maybe shouldn't be thinking about the gene that started creating abnormalities for them 30 or 40 years before they came into the clinic, because by the time they see you, they may be in a very, very different state. So we map these proteins out, and as I said, the idea is that different people live in different corners of this map, and we think of the genes as working together. So we're not doing single gene genomics and saying this gene is really important for Parkinson's disease as a standalone. We've discovered a lot of those. They exist. They're important to know about, but most of our genes remain missing from those analyses because they work together and our genetic methods are not geared to find them. So we look for these hotspots. And this is an example of one thing that we recently did. So if you start from the top right, this is a heterogeneous patient population. We sequence their genomes, and we view those genetic differences through the lens of that map that I showed you. That all genes are not created equal. Some of these genes are more likely to cause Parkinson's than others, and they fit together in a specific geometry. So we started developing machine learning methods that can diffuse around these maps and find hotspots. So we took the whole Michael J. Fox Foundation cohort, we've sequenced their genomes, which they did, and we have now started to discover hotspots in genes that then relate. On the top left, you can see to very different progression rates of those patients, or different soluble biomarkers like neurofilament light chain, or different cognitive outcomes. So we start to realize these clusters are related to something. And so this is the basic idea. Here we go. You start with these mutations in genes. It's very sparse. Sorry, this is jumping for me. And then we create these algorithms that I'm not going to go into that allow us to diffuse around these networks and say, you know what? I picked this gene out of my screen, and this blue gene out of my screen, and this brown gene out of my screen. But now I find there's actually patients that have clusters in mutations around my gene of interest. So without going into the detail, I just kind of want to convey that there are likely to be molecular subtypes. So we transferred some of this technology into company number 2, and they've gone with it now, and they've systematically looked at this, and they have found some really striking things. They have taken these very complicated progression curves from our patients. This is an example from the Fox Foundation. We've done this in 3 cohorts now, and they have converted them into genomic hotspots. So now we know that there are patients on the blue line that are traveling faster in their disease than patients on the orange line, and we've identified the hotspot in the genome that relates to it. And it turns out that this is in a metabolic pathway that I was never going to study myself. I know a couple of years ago, Michael Schwarzschild came here and talked about uric acid and how it relates to Parkinson's disease. And what we've found is that an enzyme that is related to uric acid, but it's far up in the pathway, is actually driving this change in patients. So what Michael has shown is that the lower your uric acid, the faster you progress. We've now been able to use this method to find the hotspot in the genome, a cluster of enzymes that are actually responsible for that effect. And they were not the ones that were used for the drug trial that they did, the SURE-PD drug trial, which we would predict to fail, and in fact it did. So I can't tell you we're going to succeed yet, but we have found this group of enzymes. And what's really cool is the more enzymes you have, the more mutations you have in this pathway. So this is in one enzyme. Your uric acid is a little bit lower, but then you get the more mutations you have in this pathway, the lower your uric acid. So this is an example of where we could bring in patients, we can sequence their genome now, we can say, do you have this hotspot affected or not? What's your uric acid level? We're going to now bring you into a clinical trial where we're going to target a therapy to this particular enzyme, which we know is druggable. So hopefully that makes sense. And again, it's all about finding a needle in a haystack. So I showed you this network on the left. This little metabolic pathway sits in a part of this map that I didn't even know existed. I wasn't interested, and there's hundreds of proteins here. I've had a postdoc recently spend 7 years studying a single protein in our network. So if we study 500 of these, we could be here for thousands of years, but we need to do better. We need to use computational tools to figure out, where do we even look? Where are the mutations that matter to our patients, not to a yeast cell or a human neuron or a brain organoid, but to our patients? So this is the type of biology we're trying to create. So hopefully that makes sense. Now, I mean, the point of this, I'll just brush over it, is we can, just as we did with Sue years ago in a yeast cell, chopped and changed every gene in the genome, we have now created engineered cells, this is all unpublished work, that express synuclein at the same levels as the brain, and we can make a cortical neuron, a dopamine neuron, an astrocyte, a microglia, and we can run the screens now and chop and change every gene in the human genome and ask which ones change the outcome to synuclein. So we can figure out, what does the genetic network look in neuron X, and neuron Y, and neuron Z. And so we'll get increasing resolution in these maps. This is just a visualization of how we use these CRISPR libraries now. We pool thousands and thousands of CRISPR guide RNAs in the same dish, and we can knock out genes at the same time, and we just ask, which of you neurons drop out and die before the other ones, because those are the genes that are now important in a cortical neuron. This is an example of a known Parkinson's gene, VPS35, that sits on our map. And you can see that when we delete it in neurons that are not expressing lots of synuclein, the neurons survive just fine. This is a toxicity marker. But over time, the neurons that are compromised for this protein start to die. And so this is a way we can capture a known human genetic response now in the dish and understand what its mechanism is. And this is an example of now doing this across 2,000 genes with 10,000 guide RNAs. And these are now the known Parkinson's genetic risk factors that we have captured in cortical neurons that we are now hypothesizing are related to dementia in patients. Some of you may recognize the GBA gene, which is the Gaucher protein that is known to cause dementia and cognitive decline in Parkinson's. We now have identified additional genes that should be candidates for that process. So I'm going to go through in the next, probably spend about 5 to 10 minutes now on each of these second and third sections. I know I'm going quickly. I'm happy to answer questions later, and I just want to really explain the concepts. So I've talked about genetics and how we are using models in the dish and this biology of protein misfolding to find these genetic hotspots. But of course, we're all swimming in environmental toxicants. Parkinson's disease is the fastest growing neurodegenerative disease on the planet. It is growing in prevalence beyond our aging. So something's going on, right? And I have a feeling, and there's data to support this, that the fact that we have been exposed to many toxicants that are associated with the death of dopamine or cortical neurons is probably a contributing factor. The question is how can we do that in Massachusetts, in Vermont, in California? How do we figure out which of these toxicants are really important? So this goes back to the to where we were in the 80s now, right? So we've come full circle, find the first genes. This is a very dogmatic field, so then everyone jumps on genetics and then thinks that environment, no one gets funded to study the environment for the 20 years at the NIH. And now no one's getting funded to study genetics. Everyone's getting funded again to study environment because they realize that the genetics is only explaining 30% of the disease. You know, the heritability is really 30%. So suddenly environment is in vogue. And my interest is gene environment, because I think that you have to study both, and we have cellular systems now where we could do it. The question is where do we start? And so in California, Beata Ritz and Kim Paul have an incredible study where they know for 40 years they have pound per acre exposure of different toxicants, including pesticides. And I know from talks today that you're starting to gather data like this for Vermont as well. And this is, but California have been way ahead in this, and this is just kind of a graphical visualization on the right of how different pesticides have basically increased in their usage, especially since the 1990s. So just as we have genome-wide association studies, Ritz and colleagues have performed pesticide-wide association studies, and they started teaming up with us because they say, well, we can find these pesticides, but we have no idea, are they working in the liver? Are they working in a glial cell, in a dopamine neuron? So we've started developing platforms where we can make human CNS cells and screen them against the pesticides. And this work is led by Rich, who's a terrific junior faculty member in the division, and a close collaboration also with Lee Rubin at Harvard. And so we take our similar cells on the top left, cells that have different levels of synuclein, and we basically have a robotic pipeline now where we can make dopamine neurons. We make them glow red, so we can really capture the dopamine neurons by knocking in a tyrosine hydroxylase reporter, a TD tomato reporter at the tyrosine hydroxylase locus. This means all the dopamine neurons can be purified. We can make them in bulk, and we can then start exposing them in these robots to pesticides. This is what is visualized here. This initial paper was published, about a year and a half ago. And so this is an example of propagite, a pesticide that is clearly killing these dopamine neurons. So we get dose responses, and eventually we start to figure out how are they doing this. And so we've now looked at this across all of these 50 or 60 pesticides. And what's really nice about this data is we know what co-exposures occurred in these people. Like, so these red hotspots here, these squares, are where patients in certain geographical areas got exposed to multiple pesticides. So what we can do in the dish is we can also expose the dopamine neurons to multiple pesticides and see if they line up. And that is actually really happening. And so what you see here is that there are certain pesticides. So this is actually a group of pesticides that are all used for cotton agriculture, and they're sprayed together, and we can then expose the neurons together. And what we found is towards the right side of this graph is when you start combining these cotton pesticides together, especially with one key pesticide that's called trifluralin, we really start killing dopamine neurons. So this allows us to start to think of the public health ramifications of our work and to be able to take out some specific pesticides that are synergizing with others to create highly toxic effects for our neurons. And so this is an example of trifluoralin. Every time trifluoralin is added to the combination, these neurons are much more susceptible. So we started to try and work on what is trifluoralin doing. So this is just the dose response showing the synergistic effect. We have so this is work from Edinson. We have shown that the sorry, it keeps jumping. We've shown that the mitochondrial dysfunction that I've mentioned is quite important in Parkinson's disease is absolutely triggered by trifluoralin. Other groups have shown in vitro that alpha-synuclein can aggregate in a dose-response way when exposed to trifluoralin. So there are different mechanisms through which this could be happening. One thing we're very interested in right now, the hub of our synuclein network were these yellow genes in the middle, and they were all related to calcium handling. We know that the way neurons handle calcium is incredibly important for dopamine neuron survival. There are dopamine neurons that sit in the midbrain that do not die in Parkinson's disease and die in Parkinson's disease. And one of the major differences between them is how they handle the metal calcium. And so we started wondering, do these pesticides, can we get their doses down and start to see that they're affecting calcium homeostasis? And what you see on the left here is that there's normal neurons that are firing together. These are being imaged with the GCAMP reporter. But when we put trifluoralin on at a relatively low dose, you can see that those neurons are hardly firing and their frequencies look different. And this is a pipeline we're developing and showing, for example, that the pesticide copper is completely different in the way it is affecting calcium flux than the pesticide trifluoralin. So this is something Edinson and Rich are now really working on, trying to figure out, how do we get at this, and what is the mechanism, and are there some druggable targets in this? Now, this is all growing one group of neurons from one patient at a time. This is a brutally hard thing to do. You can just ask any postdoc who does stem cell work. It's not fun. One thing we'd like to do is study an entire cohort at the time. So now we are developing technologies to take, like say, everyone in this room, make your stem cells and pool them together and grow you as a group in the same dish. Then we can start exposing you to different genetic modifiers or different pesticides, and we can start to see which of your dopamine neurons are starting to fall out with which pesticide or which genetic manipulation, and you're all in the same dish, which means that you're controlled for your environment. So this is called a village in a dish approach, and indeed we've started to do it. So we've started taking patients who progress fast and progress slow, and we're combining their stem cells, and we're starting to analyze now whether the ones who are fast progressors in life actually drop out of the dish quickly as well. Now, you may know that Parkinson's is more common in males than it is in females. So are there sex-specific differences? So in this collaboration with Alex Pollan that I'm kind of depicting here, we're taking male and female Parkinson's lines and we're growing them in the same dish, and we're looking at how they respond to different genetic and environmental manipulations. So this is an example of a dopamine neurosphere, which is a three-dimensional structure that's like a little midbrain that we grow, has the key components of glia and midbrain dopamine neurons, and but it's not from one patient, it's from multiple patients, male and female, and it's being exposed to the same pesticide I just showed you, trifluoralin, and you can see at 30 micromolar, trifluoralin is killing these cells, but at 2.5, the calcium changes are occurring, but the cells are not dying. So we're now doing single cell sequencing where we can now distinguish which came from this male, that female, this female, who's who in the dish, and who dropped out because of this trifluoralin and who survived. And what are their genetic differences? So this is how we're starting to get at gene-environment interactions in the dish. So I'm going to take you back for a minute to the family in northern Spain in Basque Country. So this family, as I mentioned, has this remarkable property where they have very different responses to the same mutation. So this is actually their full, I showed you 3 of them before, but this is now all of the patients we follow. We've been following them for 5 to 10 years with Rosario Sanchez Pernote and Juan Carlos Esteban, two amazing local neurologists. And you can see the higher these scores, the worse you are for the motor score, and the lower the scores, the worse you are for cognitive. You can see there are patients in blue and patients in burgundy. The burgundy people are doing a lot worse than the patients in blue, and yet they're the same age. They are the same age, they've seen the same mutation. So we sequenced their genomes, we made stem cells from all of them. We haven't seen anything jumping out at us from the genome. It's hard to do with like 10 or 15 patients. But we started wondering about a question that I know folks in this room are probably interested in too. What is the effect of the gut microbiome and what we eat? And is that really important? Because there are very few families in the world that have Parkinson's disease that all live in the same village, right? So they're relatively controlled for their environmental exposures, and yet they have these crazily different outcomes. So we wondered about the microbiome. So what we did was we essentially flew their poop across from Spain to Boston over years, okay? Years of poop were delivered to our lab. And we created transgenic mice that had the same mutation that the patients had. And we infused the microbiome, the poop essentially, into sterile mice that had been treated with antibiotics. And we said, are they different? So there's now a litter of mice that are litter mates. They should be genetically close to identical. And then there's effectively a kindred that's living in a village in northern Spain. And we're just trying to relate these two things with the gut microbiome and nothing else, okay, and the same mutation. And so really what's remarkable, this is just showing you the schematic. What's remarkable is we can pretty much tell who's who. Okay, the poop that comes from the patients who are really sick with Parkinson's and dementia make sick and demented mice, and those that are really healthy in life make relatively healthy mice. And so, again, without going through all the details, this is a clasping phenotype here. This is a Y maze exercise, and you can see that you should be acting like, for example, this patient A08 doesn't carry the mutation. A04 is in her 70s. She has this mutation. She looks pristine. At the same age, her cousins and her sister were demented in their 50s, and they're dying now, and she's completely fine. And you can now see just by putting her microbiome into these mice can recognize novelty in the Y maze, but her cousin A03, there's no change in the Y maze. With her gut microbiota, we're not seeing any learning that's happening here. So the same thing's happening with motor, same things with cognitive, which means there's some element of this captured in the gut microbes. This was not our original idea. The Sarkis Masmanian's lab at Caltech had a great study in Cell a few years ago where he showed this in sporadic Parkinson's, but people have really had a lot of difficulty reproducing his data. I don't think his data is wrong at all. I just think if you do this from sporadic patients, you're just going to keep finding different things, different bugs. But here in northern Spain, same mutation, same village, we think we've found the first bugs that really are associated with rapid progression, and we think that this could lead to therapeutics. What's really cool is if you look at the brains of these mice, you can also tell who's who. So, okay, so here's a healthy control looking at microglial genes, so the inflammatory cells in the brain, and here's someone who's halfway, who's got the mutation but is doing fairly well, and here's someone who's doing really badly. And you can see in the mouse brain, you can figure out who's who by just looking at the level of inflammation that's happening in the brain. So now, instead of just making neurons in the lab, we're making neurons and astrocytes and microglia so we can capture inflammation, and we're exposing these cultures to the gut, these supernatants from the bacteria, and showing very different inflammatory responses in the microglia that we're making from these patients versus other microbes. Does that make sense? So some microbes seem to be triggering this. This family is allowing us to identify what they are, and we hope that this could lead to a new generation of, probiotic type manipulations and even drugs that can be more effective. Now, I know I'm not doing that great for time, but this is kind of the last section. I'm going to blame my breakfast guests. I was the guest, but my breakfast hosts. But I want to get through this so that I think we finish at 1:15. So I'm going to try and wrap this up in the next few minutes. I'll just explain this conceptually so you'll understand what some of these slides are about, but I don't need to go through all the details. I mentioned that alpha-synuclein also twists into different shapes, right? And this we know can be quite important. If we cook alpha-synuclein in the lab and make it into fibrils and just use different salt concentrations, we can make different structures. If we put those into mice, and I didn't do this, this was a lab in Europe, we can put those different fibers into mice, they will get different diseases. They will get different diseases just based on the shape that synuclein is folded in, and we know this happens in humans as well. And so this is just a depiction with cryo-electron microscopy of how these different shapes look. But we need to start to figure out what's important about these shapes. Can we develop diagnostics and therapies that bind these different molecules? One of the things that the Alzheimer's field has that the Parkinson's field doesn't is a way to visualize the amyloid beta protein in the brain. That is why we have the first Alzheimer's drugs right now. They're not great, but we wouldn't have had a drug if we didn't know how to do it. So what I'm going to tell you is that we have now, within just 2 or 3 years, performed a small molecule screen against these different fibers, and we have put the first ones into humans. Okay, so I'm not going to show all the data, but I told you we were screening like 1 million compounds before in yeast cells. These days we can screen billions of compounds. So we screen 60 billion compounds against for molecules that would bind the alpha-synuclein fibrils and not the tau fibrils, not the beta amyloid fibrils, to start to develop new diagnostics and treatments. And this is just a scheme of what I just said, 60 billion molecules. I'm not going to go into the details. This is our chemical approach. What we wanted, and this is the take-home message, we wanted molecules that bound synuclein but did not bind A beta or tau. Because what the problem is that we can make synuclein radiotraces and they're non-specific. They start to see Alzheimer's pathologies. So we started competing these traces with thioflavin, which is where these amyloid binders bind, and we started finding new chemical scaffolds that could bind them. But the take-home message is really that we're creating a diverse library of molecules that can now bind different forms of this protein. So when a patient comes to see me now, they get 7 skin biopsies. We visualize synuclein in their skin, we grow the alpha-synuclein molecule from them, and we can put it in the dish. This is just showing that we can make these different strains. Just by taking a skin biopsy, we can capture the synuclein, and now we can put them in the patient's neurons and start to test these radiotracer candidates. And basically what you see here is that we have methods now where we can rapidly create alpha-synuclein pathologies in the dish, which is what you're seeing in green, and they can bind some of these radiotraces in a specific way. This is showing synuclein binding. This is showing that they do not bind to tau when it aggregates in the dish, so that they are specific, just like we wanted, and that they really interestingly only bind certain forms of synuclein and not others. So this is really fascinating because it means we've got a family of molecules that could bind different shapes of synuclein. So in just a couple of years, the FDA approved us to go from a screen and start putting these molecules into humans. We haven't published this yet, but the Fox Foundation tells us that this is one of the most promising molecules they've seen to date. What you're seeing on the screen here is a patient with Lewy body dementia, and that's their brainstem, and it is what we hope is aggregated alpha-synuclein in their brainstem, and there's the statistics below. There's some non-specific signal in the venous sinuses, but basically the signal gets worse as the disease severity and duration gets worse. So we think we've got our first shots on goal now for alpha-synuclein radiotraces, but we also wonder if these molecules could be a new family of therapeutics. So the final thing I'm going to really say is that we have now put all of this stuff together in a program called MyTrial in our division. And what this basically is a patient can come in, We can longitudinally track them, which is what this, what this diagram shows. We observe them, we sequence their genomes, we make a stem cell, we quantitate their biomarkers, we use biometrics. This is for a very small number of patients because it's intense. And then we give them a drug. And we ask what happens when we give them the drug. And we look and we do the drug trial in the patient, and we do the drug trial in the dish at the same time. So I'm not going to show you this data, but it was asked of me in breakfast, so I wanted to just show that we looked at brain inflammation in multiple system atrophy, which is, as you know, a very aggressive cousin of Parkinson's disease. And the take-home message is as follows. We took a drug that has failed in clinical trial, and we found that half of the patients responded beautifully and their inflammation came down, and half of them did not. And the drug company that made those drugs would not have known that outcome when they did the trial. So we are wondering if perfectly wonderful drugs are being thrown out because our patients are not responding in the way we thought. And what's really nice, and I'm going to skip past this, is that we can make the inflammatory cells from them, the microglial cells. And what you're really seeing here is what Alain has done, is shown that the responders in life are also responders in the dish, and those who do not respond in life do not respond in the dish. So we are hoping that we are building the first technologies for personalized medicine where the human stem cell models that we make will predict who responds to a drug and who does not, so we can appropriately target them. This is just a visualization of inflammation in Parkinson's disease, and we're starting our first trials like this for PD as well. A number of companies are collaborating with us, and this is my last slide. These are my take-home points, that the heterogeneity of this disease is a real barrier to understanding it and treating it. The genetic, environmental, and alpha-synuclein factors are important, that the subtypes probably matter, and we can predict now a few subtypes we think that progress faster. We are capturing inflammation and relating that to pesticides and to the gut microbiome. We're trying to capture the diversity of this protein, and we hope that the stem cell models that we make are going to be useful in ushering in a more innovative clinical trial approach in the future. That's it. Sorry I went a little over time, but I really appreciate your patience. Thank you so much.
From the in-person audience?
Thanks. That was a really thought-provoking talk. Thank you. I'm sort of sitting here wondering if I just heard a talk from 3 different people, because that seems like how many, at a minimum, it would take to do everything you just described.
Lots of good people around me.
I know, but still, it's very thought-provoking. Going back to the first section with the gene networks and the analysis of those networks, it occurred to me that, and you mentioned it in passing, that spatial distribution of the proteins in those networks could be very interesting, and with modern techniques for high-resolution proteomics, expansion microscopy, repeated labeling, you might get at that. And I was just wondering if that's sort of on your roadmap and what your thoughts are.
Yeah, the first proximity maps we did in living cells was with Alice Ting using her Apex-2 technique, which is a proximity labeling technique. That has proved to be very useful. Right now, at the Wyss Institute, they've developed a new barcoded DNA technology so that they can multiplex 15 or 20 target antibodies at the same time. And that's exactly speaking to your point. We've never been able to do that. If you're interested in that, I can put you in touch with the people who have invented that technique. It seems very good to us. What we are focusing on right now is alpha-synuclein is involved in perturbing endosomal trafficking, and they have developed a panel of antibodies across endosomal antigens, and that's going to be our first attempt to do multiplexed antibody work. Other collaborators are doing spatial transcriptomics, but not spatial proteomics yet for us. So we're doing some of these things, but proximity labeling is still something we're very interested in because we can do it in living cells.
That was an excellent talk. I have two basic technical questions. The first one is you mentioned a few CRISPR screens, one with 10,000 genes. Was your guide library selected in any way for like the druggable genome or transcription factors or any type of group?
Yeah, thanks for asking that. I glossed over it. It was 2,000 genes with 10,000 guide RNAs, and they were all known proteins and genes that we had already recovered in the synuclein network, and we wondered which is important in a cortical neuron. So they were things that we might have found in a yeast cell or a hex cell or a rodent neuron, or all these screens that we've done over the years, and we've just got a collection in the lab, and because we didn't want to do the whole genome, because that would just be this huge experiment, 80, 15 centimeter dishes to do it. So we wanted to just be more focused, but sure enough, we found that our screen did recover some interesting genes, and we think that some of those may be involved in the non-motor symptoms in PD.
Awesome. And as a fellow stem cell biologist, I found your village organoids very interesting, that you used and you coupled with single cell sequencing, but single cell sequencing isn't that deep. So how did you tell? Did you barcode your cells at all to figure out who was who and whose cells were more vulnerable than others?
So can send you the link. There's a method called Census-seq that was developed at the Broad Institute that's actually really low depth DNA sequencing, and you can actually deconvolute an entire population with just known SNPs. There's just a lot of, it doesn't take too many, it turns out, to figure out who's who. The RNA-seq is not what we're using to deconvolute. It's actually sequencing the DNA at low resolution. That tells us who drops out. But then we can sequence with RNA to then combine that and figure out what the transcriptional profile is.
Thank you.
Thank you.
Thank you. That was a great talk. I have a question regarding the environmental studies. Maybe this isn't your side of things, but how do you prove exposure? Because I've done some experiments looking at like cyanobacteria exposure and trying to link that to neurodegenerative disease. And it's just, it was really hard to prove. Like people move, and people may where they end up in a nursing home or something, which might not be the address where they were actually lived a lot of their life. So it's really hard to prove exposure. And then even are there any metabolic signatures for their possible exposures that you're able to look at?
Yeah, it's true that Kim or Rich would answer that question so much better, and I'm happy to put you in touch with them. Kim is really the person on the ground designing the study in California. What I understand is they really have a lot of details about where home is, where work is for people, which people have stuck around. A lot of them are agricultural communities too, so they actually they try and have as deep a data set as possible, and I think they've just got great records in California for this. And I think then it comes down to an association, of course, and then a mechanism, and finally finding out the metabolic signatures of exposure so that they can go into they've banked biospecimens for all of the patients, so they can now go in and find metabolomic profiles for these different exposures and confirm that they think that the exposure was correct, because they can find that evidence of that metabolic perturbation.
Do they have like controls, like family members without the
Yes.
disease that they're comparing to?
Yes,
To look for the
exactly.
same standards?
Yes, exactly, they do.
So first of all, very interesting talk. Thank you. Secondly, I don't know if I just missed this, but I was trying to figure out, have you so far, or do you plan to look into the gut biome differences and if they are affecting the protein folding strains, or if that was just associated with overall neuroinflammation being more?
That's a great question. Different people around the country and the world are looking at that in different ways. It's such an interesting question. So I think it comes down to this, that there's probably a few mechanisms that are worth looking at. What we have noticed is that there's a big change in the microglial state. So we're quite interested in how, what those metabolites might be, because we know there's a lot of gut metabolites that get into the blood and can affect it. So that's one aspect of it. The second aspect that one of my colleagues at University College London, Soyeon Hong is working on. She's done beautiful work. It's not published yet, but she's done some really amazing work showing that there's triggering of immune responses in the gut, and that the T cells that are activated in the gut actually get into the brain. She can demonstrate that because she labels them. Same ones that get activated here end up going in the brain, turning microglia on and creating inflammation. So that's the second. And the third one that you're getting at is, are there strains of bacteria in which they can actually trigger misfolding of the protein? And yes, I mean, I can't remember his first name, but last name, I think it's Tim Sampson. His lab is looking at a particular type of E. coli that has a protein called curly that can actually co-aggregate with synuclein and seems to be able to trigger alpha-synuclein aggregation in the gut wall in enteroendocrine cells, and the synuclein then passes to neurons and can travel through the vagus nerve to the brain. So that's pretty interesting. Yeah, I think those are the three main mechanisms that I'm thinking of.
I'm going to ask one question too. So, you mentioned like there might be a lot of drugs we are throwing out because of the approach we're taking, we're not acknowledging the heterogeneity of patient population. I'm wondering if you can comment on the recent sort of the alpha-synuclein antibody trials which have failed in light of your work. Like this, do you have a comment of why they might have failed in view, like their targeting of, are they again the same reason, something more just more fine?
Yeah, it's really interesting. So, firstly, failure is okay, and maybe they'll succeed soon. We've seen that happen in the Alzheimer's field. At many levels, I think. The first is, as Roche and Prothena have pointed out themselves, their patients were so heterogeneous that they were on different progression trajectories, and they had a lot of noise in their study because some people were not progressing. And so at a simple level, they might have had a drug that worked, but had just too many people with very heterogeneous progression curves. So our findings could help that because we might be able to enrich for patients who are progressing faster based on their genetics. And so that's something that we're interested in. Maybe it's just that. But there are other biological reasons why this, I mean, I love synuclein, but as I mentioned, when synuclein misfolds, it's doing it decades probably before the patient comes in. And I think there are a lot of consequences within the cell and outside the cell that we need to take into account once a patient is progressing. The biology of synuclein may be incredibly important for risk, maybe even the onset of the disease, but not necessarily for progression. And that's why I feel we should be looking at what synuclein does a little more globally, because some of the druggable targets may be there. It's an important protein, but it's worth thinking about its implications. And finally, when alpha-synuclein aggregates, it mislocalizes. It finds itself in a place where it shouldn't be. And so a disease that starts, let's say simplistically in the woman I showed with too much synuclein, by the time she's progressing in a lot of her neurons, there may be too little synuclein in the place where it should be. And so we need to be really careful. We were talking about Huntington's this morning. We need to be really careful when we knock down genes or use antibodies of whether we're disrupting their normal functions and actually starting to exacerbate some problems that are more likely to be problems in the aggressive phase of the disease. And I think one of the biggest challenges we need to face is that the same brain may be in different stages of the disease at the same time. That a dopamine neuron that's dying with neuroinflammation around it may be a different therapy than a cortical neuron that is succumbing and is still in its early stages of degeneration. So we have to start thinking about that and modeling these different processes.
Well, thank you for that wonderful talk. Very, very enlightening.
Thank you for inviting me.
And thank you all for coming in person and virtually. We'll close at this point. Thank you.
Thank you.